Have you ever really looked at the food on your plate and just wondered, how? How did it get to be so perfect, so delicious, so abundant? Well, the story behind it is way more incredible than you might think. It's a 10,000-year-long saga of discovery, of incredible ingenuity, and, yeah, of humanity learning to control the very building blocks of life. So let's pull back the curtain and uncover the secret history of what we eat.
All right, to get this, we gotta set the scene. Let's go back to 1955. The entire world was home to about 2.7 billion people. Now that's a huge number for sure, but what happened next was just staggering.
Because just a little over 50 years later, by 2011, that number had more than doubled to over 7 billion. I mean, just stop and think about that for a second. We had to figure out how to feed twice as many people all within a single lifetime. It was, without a doubt, one of the greatest challenges our species has ever faced. And to meet that unbelievable challenge, our food production had to do something just as dramatic. Our crop yields, that's the amount of food we can grow on the same piece of land, they had to just absolutely skyrocket to keep up. But here's the thing, this wasn't an accident. It wasn't magic. It was the result of intentional, brilliant design. So the big question is, how on earth did we pull it off? Well, the answer starts with understanding the absolute fundamentals, the basic rules of nature. See, before we could even think about directing the show, we had to learn how all the actors behaved, and that meant getting up close and personal with how plants, make more plants. So let's quickly meet the key players in this little drama. You've got the anther, and its job is to produce the pollen. That's the male contribution. Then you've got the stigma, which is like a perfect little landing pad, designed to catch that pollen. And then deep inside, tucked away safely, is the ovule, which holds the female egg cell. And that incredible moment of transfer, when a grain of pollen lands on the stigma, that's what we call pollination. It's the essential first step. It's like the handshake that kicks off the whole process of creating a brand new seed. But honestly, the really mind-blowing part happens next. Inside that little pollen grain are two sperm. And what happens is this brilliant, super efficient process called double fertilization. One sperm goes and fertilizes the egg to create the embryo. That's the baby plant, right? But the second sperm, it goes and fertilizes other cells to create something called the endosperm. And the endosperm is basically a packed lunch for the embryo. It's the food supply that's going to sustain it until it can grow its own roots and leaves. Nothing is wasted. Understanding this elegant little blueprint, well, that was the key that unlocked everything for us. Because once we understood the mechanics, that's when the long, incredible journey of human intervention really began. We went from just being passive observers of nature to being active designers, literally shaping the plants that feed the world. It all started about 10,000 years ago with simple domestication, just farmers saving the seeds from their best plants. But then our understanding just exploded. The rediscovery of Mendel's laws gave us the rulebook for genetics. The discovery of DNA's structure in '53, that gave us the source code itself. And all that knowledge led to the Green Revolution in the 50s and 60s, this incredible burst of innovation that totally supercharged our crops. And now we're in the age of precise, direct genome editing. And believe me, this was no small change. The impact was so profound that Norman Borlaug, who we call the father of the Green Revolution, was awarded the Nobel Peace Prize. The committee itself said that this new technology had saved potentially 1 billion people from starvation. I mean, this was science directly shaping the destiny of humanity. So, armed with all this incredible knowledge, plant breeders came up with some really clever techniques, biological hacks really, to take even more precise control over how plants reproduce. It was all about designing that perfect crop. One of the first things they had to master was one of nature's own systems, something called self-incompatibility. Think of it like a genetic firewall. It's a built-in mechanism that stops a plant from pollinating itself. And you might ask, why? Well, it's all about ensuring genetic diversity. By forcing a plant to cross-pollinate with another one, nature guarantees a healthier, more resilient gene pool. And this isn't some rare, weird thing. It's actually super common in a lot of the foods we love, like apples, cherries, and even cabbages. It means for these guys, you can't just have one lonely apple tree and expect fruit. It needs a genetically different partner nearby for pollination to even happen. So breeders had to get this rule down cold to be successful. But that raises a really big question for a breeder, right? Self-incompatibility is awesome for nature's grand plan. But what if you, as the designer, want to create a very specific hybrid? What if you want to guarantee that pollen from this exact plant fertilizes that exact plant, and nobody else gets involved? How do you force that cross? This is where breeders get really clever. They use a natural trait called male sterility. Basically, they find a mother plant that physically cannot produce its own pollen, so there's zero chance she can pollinate herself. Then they just bring in the pollen they want from the specific father plant they've chosen with all the best traits. The result? Every single seed that's produced is a guaranteed 100% pure hybrid of those two parents. It's a genius biological hack that gives breeders complete control and takes all the guesswork out of the equation.
And that, of course, brings us all the way to today, because this story is far from over. In fact, our ability to design our food has taken another massive leap, moving from the level of the whole plant right down to the individual letters of its DNA.
I mean, just look at the modern toolkit. It's like something out of science fiction. We've got tools that let us find the exact genes we want almost instantly. We've got genome editing techniques that let us make incredibly precise changes directly to the DNA. We're even using drones to fly over fields and analyze plant health from the sky. Work that used to take an entire career, decades, can now be done in just a few short years. It's a whole new ballgame.
So, what does this all really mean? It means we've fundamentally changed our relationship with the food we grow. We've moved beyond just being shepherds of plant genetics, guiding them along. We are now the architects. We are no longer just crossing two plants and hoping for the best outcome. We are actively, intentionally writing and rewriting their genetic code.
And that leaves us with a pretty profound final thought to chew on. For 10,000 years, we have been shaping the life that sustains us, turning a wild grass into modern corn, a bitter root into a sweet carrot. Now, for the very first time in history, we have the power to edit that life, gene by single gene. The question is no longer if we can do it. The question is, what should we design next?